Good morning and welcome to our MedCity and Best Precision Medicine Conference. I'm Stephanie Baum, Director of Special Projects with MedCity News. This session kicking off our conference focuses on pediatrics and precision medicine. In 2020, the FDA approved 7 personalized medicines for rare genetic diseases, according to data from the Personalized Medicine Coalition. Many of these therapies are designed to address or even reverse the genetic root causes of illnesses that begin causing symptoms in childhood. Personalized medicine is also changing the way children are diagnosed and treated for suspected rare genetic diseases. Comprehensive genomic sequencing is showing promise as a more efficient replacement for the battery of tests patients are given to arrive at a diagnosis. To discuss these and related issues in pediatrics, we have a group of industry experts from 3 pediatric hospitals. Moderating the discussion is Dr. Sam Blackman, co-founder and chief medical officer of Day One Biopharmaceuticals. Before starting his own company, Dr. Blackman was head of clinical development at Mavu Pharma, a drug discovery company focused on leveraging the immune system to treat cancer and infectious diseases. Previous roles include head of clinical development at Silverback Therapeutics and senior medical director at Juno Therapeutics. Prior to that, he held roles of increasing responsibility at Seattle Genetics, Merck, and GlaxoSmithKline. Welcome to the panel, Sam. Also on the panel, we have Dr. Josh Bonkowsky, a pediatric neurologist specializing in clinical care and research. He is a professor of pediatrics at the University of Utah School of Medicine. A physician scientist, Dr. Bonkowsky studies nervous system development and disease and the role of genetics. He has been continuously funded by the NIH since 2006, including the 2012 Director's New Innovator Grant. He's the Division Chief of Pediatric Neurology at Primary Children's Hospital and the Director of Primary Children's Center for Personalized Medicine. Welcome to the panel, Josh. Also on the panel, we have Dr. Tomi Pastinen, Director of Genomic Medicine Center for Children's Mercy, Kansas City. He's been at this institution since 2017. And there he leads Genomic Answers for Kids, a foundational initiative shaping genomic medicine that aims to learn from 30,000 patients and their family members how to solve unsolved rare disease in children. He joined Children's Mercy from McGill University, where he co-led the Genome Center and supervised a multidisciplinary human genomics team from 2006 to 2017 as a professor of human genetics. Welcome to the panel, Dr. Pastinen. And finally, we have Peter White, Senior Director of the Steve and Cindy Rasmussen Institute for Genomic Medicine at Nationwide Children's Hospital. A genomic scientist and innovator, Peter's driven to create a future where no child with genetic disease goes undiagnosed and every child with cancer receives optimal treatment based on their personal genome sequence. He directs the Computational Genomics Group at Nationwide Children's Hospital, and he has substantial experience in cloud computing and its applications in a combined healthcare and research setting. He's also a tenured associate professor at pediatrics at the Ohio State University School of Medicine. Welcome to the panel, Peter. And before I hand it over to Sam, just a reminder that the audience is encouraged to ask questions, which you can place in the chat. And now I'll hand it over to you, Sam.
Great. Well, thank you so much, Stephanie, and thank you very much to the MedCity News team for inviting me to participate in this, I think, really special opening panel for your MedCity and Best Precision Medicine meeting. As Stephanie mentioned, I am a pediatric oncologist and physician scientist, and thrilled to be accompanied by Drs. White, Bonkowsky, and Pastinen to discuss pediatrics and precision medicine as part of this panel. I do love the idea of this conference starting with pediatrics. I think that as a pediatric oncologist and somebody focused on the health of children, this is an area that oftentimes goes overlooked in drug development and therapeutic development and diagnostic development. Mainly because we think of children as healthy, but for those of us who've worked in this area for many years, as the panelists all have, we know that devastating diseases do happen to children. They happen far more frequently than I think any of us would imagine or like. And it is our job and our responsibility to create new therapeutics, new diagnostic modalities, and new treatment alternatives for these families. So really looking forward to diving in on the work that's being done by our various panelists. And what I'd like to do to begin the panel is really ask each of the panelists to take a couple of minutes and describe their program briefly, the focus of their work, and really, how they think about the burning questions or cutting edge topics within the broader field of pediatrics and precision medicine. So I'm going to just go in alphabetical order by first name. So Josh, if it's okay, if we start with you, I'd love to hear about what is the focus of your work and your program, and really what you see as the key issues in pediatrics and precision medicine today.
Thanks, Sam. Thanks for the chance to be on this panel and to meet everybody today. So I work at Primary Children's Hospital, which is in Salt Lake City, Utah. It's the children's hospital, in fact, the only children's hospital in Utah, and also kind of across the region. And we actually end up covering a large referral area, about 400,000 square miles, with no other pediatric subspecialists, which means we just have a kind of a large patient population that we take care of. It ends up being about 2 million children that kind of funnel into our center. And so what we've done over the past 5 years really is launch this Center for Personalized Medicine at Primary Children's Hospital. And we're fortunate because Primary Children's Hospital is part of this healthcare network called Intermountain Healthcare, which partners with the academic institution here at the University of Utah. And so the three big things we've worked on over the past couple of years, one is using this technology that I'm sure many people are familiar with called rapid whole genome sequencing, which is the complete diagnostic genetic testing, kind of the most advanced test available. It's been pioneered at Kansas City and then now also from Rady Children's Hospital, but now we brought it in-house here, but then we're applying across the network of hospitals here in Utah, which is kind of transforming our diagnostic ability. So that's been a huge game changer. The second big thing we've been working on is using the fact that now we're diagnosing people and children and trying to get therapies to them. So we have a big clinical program in gene therapy, and then we also have research and translational programs in new therapies for children who have otherwise untreatable diseases. And so that's been really exciting to see things go from being not even diagnosable to being treatable. And then the third big thing we've been working on is we have a new program called the Heredogene Children's Study, which is doing genetic mapping for 50,000 kids. So it's the biggest genome mapping project of its kind in the world for pediatrics. And so that just launched this past year. And because we have, essentially complete ascertainment of all the patients and children we see, it gives us kind of this unparalleled ability to understand health and disease in a pediatric population and kind of match that with the genetic information that we're getting. So those are kind of the three big programs we have here at Primary Children's Hospital. I think you asked briefly, what's the things we see, kind of looking towards the future. And I think we had kind of tossed around this idea in some preliminary discussions before when we were kind of meeting before the seminar today. And this idea that we're moving into a post-diagnostics world. So in just the 15 years I've worked as a pediatric neurologist, we've gone from where we know someone has broadly speaking, some sort of disease like seizures or epilepsy, but not being able to say anything more concrete than that, to now where we can make a diagnosis in, more than, kind of approaching nearly all the children that we take care of. So that technology is now here clinically. We're using it, every day. So the challenge that we're moving into is how do we deliver therapies? How do we develop therapies for the thousands of different illnesses? How do we personalize them? Because different types of genetic mutations need different types of treatment, even for the same gene. And then how do we make sure that we get it to our patients? Because if you live in a rural area, if you live in an inner city population area, if you don't get diagnosed correctly, even with these technologies, we're not getting them to the people, it doesn't really make a difference. So those are kind of the challenges that we're thinking about for the next decade.
Great, thanks, Josh. That was absolutely terrific. Peter, if I can put the same type of question to you, that'd be terrific. We'd love to hear about what's going on at Nationwide Children's.
Yeah, sure. So I've been at Children's, this will be my 13th year now. And I've been working as a genomic scientist there since over those last 13 years. And it was about, similar to Josh, it was about 5 years ago that our hospital leadership recognized the impact that genomics was going to have in the area of precision medicine and in pediatric care, and made the commitment to form a new institute, the Institute for Genomic Medicine. So that was formed in the fall of 2016. And really over the last 5 years, we've been working towards that goal of creating a future where no child with a genetic disease is going undiagnosed, and also focusing on an area in cancer, so that ensuring that every child with cancer is receiving an optimized treatment based on the specific somatic mutations that their genome might have. So we've become, we've really grown into a large operation over those last 5 years. We have over 150 staff and 16 faculty with research interests in that encompass genetic disorders, rare or as yet to be diagnosed diseases, pediatric cancers, epilepsy, congenital heart disease, birth defects, and we're expanding now into behavioral health. We also have an interest in technology development and the application of different genome sequencing technologies and how they can be utilized eventually in clinical care. We also have a large CAP clear certified clinical molecular diagnostics lab, and they've been providing multiple molecular diagnostics, including exome sequencing, targeted fusions, gene fusion panels, and methylation profiling. And we see that those operations shifting towards genome sequencing. So in terms of some of the challenges we've faced as we've set up this institute and as we look to the future, I think initially one of the goals we set out to do was blur the lines between clinical and research operations. And that's quite challenging because there's different mindsets in terms of the standards that are needed when a clinician's treating a patient. The standards are obviously very high in terms of the genomic data that we're generating and what the results actually mean that we're able to determine. It's been challenging, but we've done quite well in that regard, but there's still progress to be made. The second area is my specific role in IGM is leading the computational genomics group. We support all of the research and clinical informatics needs of the institute and its faculty. Back in 2016, we made the decision that we would have all of our computing based in the cloud. We've had to overcome a lot of challenges around HIPAA and other regulatory requirements, and working with a very conservative hospital IT department. And it's been going well for us, but our challenge has been in terms of growth of data. I think at the start of 2020, we had 3 petabytes. We're now at 6 petabytes, and I think it's growing exponentially. I think that will double in the next year as we have 3 of the free of Illumina sequences that each is capable of generating 50 human genomes every 2 days. So managing and organizing all of that data is becoming a challenge, and the cost of all of that data is becoming is significant. And then finally, the third challenge I was thinking about was one of our biggest needs is around the area of automation of interpretation of genomic results. We've done a great job in terms of taking data from a sequencer, or say all the way through to primary and secondary data analysis. But there's still a challenge around, okay, what does this genetic variant mean? Is this disease causal? Or in the case of cancer, how is this going to impact clinical care? And so looking at ways to automate that interpretation process using technologies like machine learning or artificial intelligence are some of the goals that we have over the coming years. So that's it for what I would say is our 3 main challenges.
Excellent. That's terrific. Thank you very much, Peter. And then Tomi, certainly to wrap up this introductory part before we get into questions, very keen to hear about the program at Children's Mercy in Kansas City.
Hi, everyone, and thanks for giving the opportunity to present some of our work and discuss with the experts. So what we do here at Kansas City is as we heard from the previous speakers, Children's Mercy Kansas City has pioneered some of the rapid sequencing in the past, and actually when I joined 2017, there was already a very flourishing genomic sequencing lab here, which pioneered the rapid sequencing, but also carried out a lot of genetic testing in a non-rapid fashion for standard genetic diagnostics. And what surprised me, coming from a background in human genomics, and I originally started 25 years ago in a Mendelian genetics lab, was that there was still majority of kids not receiving diagnosis. So even though Dr. Bonkowsky mentioned that we will move into a post-diagnostic world, unfortunately, that's not the case for a lot of kids in a diagnostic odyssey, and the families that are frustrated of not understanding what's wrong with disease. So I saw it as a great challenge that we had about 30 to 40% plateau in providing molecular diagnosis when a physician has suspected a rare genetic disorder. So understanding where does the remaining molecular diagnosis is, we decided to pursue a very large scale genomic answer for kids program that is kind of an extension of the undiagnosed disease network programs, but really trying to collect all those undiagnosed cases regionally and even beyond as we move through our phases. And over the next 6 years or so, we aim to grow the case load of undiagnosed disease from the current 7,000 that we have in our database to close to 30,000. And we've already collected around 8,000 patients and family members in our study. And this is steadily increasing. And in addition to collecting these patients into this data resource, we are generating, beyond the clinical sequencing, additional technologies to try to solve the unsolved disease, using technologies, but also using interpretation techniques that are beyond sequencing alone. So I think there will be an era where we'll see that the DNA itself becomes unreadable unless you have other molecular information from the patient, and collecting information on their transcriptomes, on how their epigenome is behaving, and how the individual cells are functioning, which can all be done actually by current sequencing technologies. One of the focuses of kind of post-sequence interpretation of individual patients' genome. And it's all geared towards more comprehensive precision medicine, because if we want to get to that post-diagnostic era and therapy development, I think we need to understand all the patients at molecular level and group them into mechanisms that may be targetable by common therapies, and not solely by targeted therapy to single disease. So our challenges here are, like I said, developing these technologies that are going beyond short read sequencing and whole genome sequencing, including long read sequencing, and scaling that up, looking at the human genome beyond the reference genome framework that everyone knows has gaps in it and is inadequate to describe the whole human genome. So building personalized assemblies of human genome, and how would you scale that up in a clinical manner? The second challenge beyond the technologies and interpretation of DNA, what we see is data sharing. So we do have a large number of labs, including the ones that we just heard in hospital settings, but also in the commercial settings. And all this data is being siloed in different programs, and we are not really seeing what's happening at Intermountain Health or in Columbus, Ohio, let alone some companies like Invitae. And actually, the patients would benefit most if we would be able to share this data, put it together, and start to interpret the uninterpretable DNA in these rare cases through comprehensive data sharing. So we are trying to make our data repository completely shareable to investigators in the field and clinicians alike. And the last challenge that we're tackling with and have observed in our early phase of the Genomic Cancer for Kids program is that the underserved minorities are still being really underserved in terms of molecular diagnostics. So how do we get out there into the community and help the families that don't necessarily even have a primary care provider? And that's going to be a great challenge over the next couple of years. And we're launching pilot programs into trying to give genomic medicine abilities directly to primary providers in the primary care clinics around inner city here in Kansas City.
Thank you for that. So I really appreciate everybody providing their background and their focus on what their current focus is, and importantly the challenges. There's actually so much here to talk about. No way that we'll fit it all in 30 minutes. I'm going to try and touch on, I think, some of the hopefully the more interesting topics for a general audience. And again, would encourage people in the audience to please put questions in the chat, and we'll make sure to leave time along the way and certainly at the end to take some of those questions. But what I'd love to do is start with as an oncologist, precision medicine approaches to both adult and pediatric oncology, I think, are pretty well accepted. It's certainly what I would say, the general public, the FDA, investors, therapeutics companies are familiar with when they think about precision medicine. It's typically cancer. There's an actionable mutation. If it's in a kinase, you make an inhibitor, and the development path, I think, is relatively straightforward. I think that is made possible by the fact that driver mutations in cancer are we've been studying them for a long period of time. We understand how to translate from the mutation back to the underlying biology to try to assign, to the best of our ability, causality, and then target therapeutic development. I do have a question. I'll sort of leave it to whoever wants to answer first. Is as you're sequencing children with suspected genetic disorders, and you're discovering sequence variations that may or may not be that may be novel or may not necessarily be. It may not necessarily be intuitive what the mechanism is. Who is doing the back translational work to go from that, or the translational work to go from that mutation to biology to get to, away from just correlation to causation? Because if we are, in fact, moving into a post-diagnostic world, we need to think about therapeutic development. Understanding that fundamental biology is going to be critical. So, I'd love to hear, and maybe I'll just pick on Peter, sort of in the middle of my screen here, to tell me, how are we set for that type of translational work, and is it going to be able to scale with our ability to generate those sequence variations?
I think you started that question in terms of cancer, but are you thinking more in terms of constitutional disease?
Absolutely, I'm thinking about germline disease. I think cancer. Okay. We have great models for cancer. Maybe it's easier to get cancer cell lines. It's more costly to make genetic models of germline disease.
Right. And so for us, our clinical, so in terms of the clinical, the clinical reporting of causative variants is very much largely linked to, hey, this is previously reported. And for patients where there is no variant that's found that fits it with a previous clinical report, they kind of shift into our rare diseases program, where we'll perhaps take a whole genome approach to sequencing rather than exome sequencing. There we're sort of seeing, I think Tomi mentioned this, the yield is around like 30% with our clinical exomes. We can take that up to 40% if we do genomes. And we're often in for the rare disease patients, we'll find those variants. It's like, hey, this is a de novo variant, there's some literature out there that supports that this could play a role in that patient's disease. And our strategy has really been to try to find collaborators that either have worked on that gene or work on similar gene families, and reach out to them to see if we can engage them in collaborative work. But that's obviously not scalable. That's been a challenge for us. And so I think some of the technologies that we've been looking at have been like looking at like different iPS cell based therapy, like looking at iPS cell techniques to model those genetic variants, looking at organoids. We have a faculty member that specializes in like creating, he's focused mainly on brain organoids, and then looking at some of the mutations that we've seen in our epilepsy patients and seeing if we can confirm that in an organoid model. But again, that's a very slow process and can often turn into years of work.
So that raises, I think, a really interesting, sort of additional challenge. And maybe Tomi or Josh wants to comment on this. An additional challenge, I think it's evident on its face, right, that rare disease therapeutic development can be challenging to get funding for or to get industry interest in because it's rare. Certainly there's a model right now that affords for rare disease therapeutic development, but it is not the majority of drug development. So one challenge is getting interest and funding for that. Obviously, the others are translational models. I'm curious to hear your thoughts as we identify more mutations, or we can identify patients with rare diseases. You've now been able to put a mutation on a phenotype, or assign a mutation to a phenotype. How do you think we're going to be able to more effectively turn some of those findings into therapeutics for patients? And if we can't, I'd certainly love to hear you comment on then what's the benefit? And we discussed this before, but I think you said a lot more eloquently than I can. What the benefit is of diagnosis without necessarily a path to a therapeutic. But let's start with the potential for therapeutic development, and maybe Tomi or Josh, either of you.
So I can start. I think Josh is better to conclude with the more positive note, but I think what I see is that the low hanging fruit are relatively rare in rare disease. So that would be gain of function mutations that we could interfere, probably conditions that don't start in utero, that are not early in development. So there's a lot of barriers to effective therapies just from the basic pathogenesis view of disease and their very early onset. But certainly, if we put our effort into kind of generic ways of for example, silencing overactive genes and gain of function mutations, that's probably the easiest way forward. But there's a lot of heavy lifting then for majority of rare disease cases where we absolutely don't have a strategy to interfere with the causal mechanism, but perhaps with greater understanding, developing those cell models, developing the animal models, we can mitigate some of the progression of the disease. And I think that's the overall hope for the future for many of the rare diseases, is to slow them down, and even if you're not able to treat the primary defect.
Yeah, I think that's a great point, Tomi. Just thinking about it, I think the classic model that we've hoped for as investigators and clinicians and as investors is that, let's get a home run, and cure the disease all in one fell swoop. So I think like the spinal muscular atrophy story is kind of that example. But if you think about the cancer story, especially in pediatrics, it's not a single home run, but it's a series of incremental gains. And I imagine that we should be thinking that for all these diseases, that if you can kind of chip away at the disease phenotype, that may be what we're really looking at over the next 10 or 20 years. The second point I'd make is that right now we're kind of stumbling around in the dark, in a foggy, dark place where we can't kind of see the scope of disease. And it could be that as we understand things better diagnostically, the fog kind of clears and we can say, okay, even though these diseases and genes are different genes and diseases, they start to share these common pathways of pathology, and that gives us some pressure points for therapy development. So maybe we can be more hopeful about some aspects in the future where we get a bigger understanding of the kind of genotype phenotype landscape.
That's really helpful. It does raise another interesting question, though, right? The idea of chipping away at diseases, to some extent requires a sense of what it is that you're chipping away at over time, right? Particularly if you're talking about slowing disease progression, you need to have a pretty good sense of what the baseline state is for disease progression, and that raises another information question. I'd love to hear your assessment of where we are today. Obviously, natural history of these diseases over time, understanding things. When I think about this from the point of view of a therapeutics developer, what are the outputs that we can measure? How can we observe change over time? What are the endpoints, for example, of some therapeutic intervention? Thinking from the point of view of not only regulatory authorities, but things that are real clinical benefit in the eyes of parents or patients. With cancer, again, the low-hanging fruit, it's easy. The tumor grows, the tumor doesn't grow, the tumor shrinks. Patients live longer or they don't. But for some of these, for other diseases, seizure disorders, unusual cardiac diseases, behavioral, somebody had raised, the potential of sequencing patients with unclassifiable behavioral disorders. The natural history part is going to be important. So how is it that natural history data for these diseases as they're identified is correlated so that you really have a fuller understanding of that genotype-phenotype relationship? And maybe, Tomi, I'll start with you.
Well, I think that's an extremely good point, and everyone who works with genetic diseases knows that even within a family there can be high variation in manifestations of the same mutation. So it is a great challenge, and especially with the low numbers of each of these diseases, building those natural histories to a level that would please FDA or your standard drug developer may not ever happen for some of these diseases. But I would kind of turn it around for understanding the architecture of genetic disease differently. So we do have a lot of diseases that have a common diagnostic label like autism spectrum disease. And actually autism spectrum disease encompasses large number of rare genetic diseases, but then the more garden variety that we don't have any high impact genetic variation. But what if you would target autism spectrum disease interventions differently to the patients that you have a molecular diagnosis and the patients that is the more kind of population form of autism? I think that could make in understanding how to start to use precision in drug trials and therapeutic interventions more tangible for us, rather than starting from the most difficult part, which is the disease that occurs in 1 in 100,000 individuals, and you only can gain a handful of patients into your trial.
I think, sorry, Sam, but I thought a point that Tomi brings up is that with the patients with the same genetic disease, but with different phenotypes, the key aspect that's going to be critical will be kind of linking the genetic information back to the clinical information. So I think trying to figure out how either family and patient organizations or NIH or different groups can develop understanding of disease phenotypes over time is going to be super critical to kind of targeting which thing you can try and treat with which therapy, or who are you diagnosing or who you're not diagnosing. So I think these kind of large scale collaborations are going to be super critical for moving this forward.
Yeah, no, I would absolutely agree with that. I think what we're faced with now, it's like a very exciting opportunity in terms of, hey, we can generate all of this data on each patient at the genome level, and then looking at RNA expression profiling and then epigenetics. But being able to have that data in a way that's connected with that patient's electronic healthcare record, and being able to like mine and look for connections between, okay, this is what we've seen in this patient in terms of their clinical care and their response to different drugs, and how that might tie in with their genomics. But in the case of pediatrics and rare diseases and even pediatric cancers, these patients are often an N of 1. And so it's critical to come up with ways in which we can share this data and across multiple organizations.
I couldn't agree with you more. And I do want to come back to the sort of the data sharing issue, but I want to maybe stick to the patient level for just a couple of moments, because at the end of the day, even in many instances, and I think somebody mentioned this in the introductory remarks, we don't necessarily have a therapeutic intervention. And what we're talking about is quite a bit of work, not just generating the data, storing it, analyzing it, sharing it, correlating it with phenotypic data, diagnostic data, radiographic imaging data, and the like. It's expensive. And so this raises the question of funding, and I think it's appropriate, because I think for this conference we certainly have people who are potentially investors or people representing industry. This is clearly important work at a patient level, and maybe just for those of you who are at the interface of how this information makes a difference to families of, in this case, children with genetic disorders, I'd love to hear your insights into the benefit of providing this information to patients, and not just the psychological benefit. I think somebody said in our pre-meeting that it helps resolve the agony of not knowing, but maybe the more tangible benefits to the system in terms of how the sequencing information changes the trajectory of the diagnostic quest, to the diagnostic odyssey. And maybe I'll start with Josh here.
Yeah, that's been I feel like I'm going to repeat what I tell the insurance companies, but for diagnosis with these new diagnostic tools, there's kind of 3 things that we tell families for why diagnosis is important. I mean, usually they're already convinced, but I think the first is that in a small number of cases, there is the option for therapy. So, one of the great examples, we had a kid who kept having seizures. They had they're in and out of the hospital like 30 times in the first 3 years of life. They had several $1 million in hospital costs. They already had some less sophisticated genetic testing, but then with a whole genome sequencing, we found that they had a treatable genetic epilepsy, and we treated them like basically the next day the seizure stopped, and they haven't been back in the hospital. So of course there's like a small number of patients where the therapy really makes a difference. The second thing, as you mentioned, is the psychological aspect, that the families for these disorders are fighting this unknown enemy, and they can't tell their uncle, like, what does their kid have? They can't be on Facebook groups for the disease. They can't kind of reach out to understand, like, in 5 years, what's the future going to look like? So it really changes their landscape of the future. And then the third thing is that even in cases where the diagnosis is fairly grim, and even like can lead to withdrawing care for a medically complex child, I think that brings an element of peace to the family. We've had some cases where we've used genetic testing and the families decided to withdraw care because the pathway forward is futile with fairly grim medical interventions necessary, but knowing that they've done everything they can, that they know what they've faced, and then making the decision that in the best interest of the child that they should withdraw care, has made it, we've gotten thank you letters from families for that kind of diagnosis. So there's really options for the diagnosis makes a huge difference.
Tomi, Peter, any thoughts from your experience here?
I think that was really elegantly put, and these are really the points that that are raised. And we actually, in our genomic medicine program, we have in the research side, we have patient advocacy groups and families with rare disease participating in our meetings and really echoing what we just heard. It's always important to get the diagnosis, and in most cases, the sequencing is the end point of reaching the kind of final goal of getting the diagnosis, but often what these families are faced with is multiple layers of medical care where no one believes that there's something odd with their kid and that something more needs to be done. And I think that's one part that we need to be more conscious of is the barriers to getting referred to genomic medicine, because the number of genetic testing we do in a tertiary care hospital like ours that covers much of the 5 million people population base, we know that there's many families out there that would need genomic testing but are not offered that.
Just a question for you in this way. What I think or what I'm hearing in this is, early arrival at diagnosis, shortening that diagnostic odyssey, and particularly making it far less complicated and far more credible, by employing next generation sequencing and other genomic technologies. Has a strong potential to reduce costs of the system, because you're not bouncing from clinic to clinic, constantly in search of a solution. I think about all of the, the endless, standard laboratory tests or imaging studies or other diagnostic interventions or diagnostic modalities that are employed when there's no one clear unifying theory. So that cost savings, seems to me at least to be prima facie pretty apparent. But that's a health economics and outcomes research problem, something that could be demonstrated. It sounds like there's a broader research program that could potentially address this, looking at the time and cost to get to somebody to diagnosis 10 years ago versus the time and cost to get to somebody to diagnosis now. And at the end of the day, we're going to have to convince payers to reimburse, or I'd love to hear how you're funding patients who can't necessarily afford sequencing. But maybe a two-part question. How are you paying for this now, or how is this being paid for now, and who should pay for it going forward, and how do we convince them that at the end of the day it's cost effective? Maybe I'll start with Peter.
Yeah, sure. So I think that's a great question, Sam. So I think for the case for patients with rare genetic disorders, I think the evidence is pretty clear that, hey, these technologies can end up saving like the insurance companies a significant costs because of ending those diagnostic odysseys, and there's so many examples of that. I was thinking more in terms of like some of the work that we've been doing for patients with rare or refractory cancers. That so far we've been taking an approach of enrolling those patients into a research trial that's funded by philanthropic sources through the Children's Foundation. And with those kids, we've sequenced about, I think, just over 350 kids now, where it's been initially a research focus to show the potential impact of sequencing the tumor genome from these patients. For many of these kids, they haven't responded to the standard of care. And then when they go through the sequencing program, it's like, okay, there is a molecular change that's clearly targetable. This therapy that targets that change should have been like the frontline approach in this patient's case. So I think the shift that we'll see is like we've been able to demonstrate that, and many other groups are demonstrating it as well, that okay, the findings that we can find by using these technologies will change healthcare, like will change how the patient's cared for clinically, and ultimately like that's better for the patient in terms of like hopefully their cancer will respond to the treatment better, and then it's better for the third-party payers because their costs of that hospital care will go down. So we're seeing a shift. Initially, like in terms of funding, initially it's been largely philanthropic, but there absolutely needs to be that shift, having proven the therapeutic benefits of these approaches.
Tomi, I see you nodding in agreement here.
Yeah, so similar to what Peter was mentioning, most of the support we have for large-scale genome sequencing, either in cancer or mostly in genetic rare diseases, is philanthropic currently. There hasn't been, there are few programs, NIH-funded programs that fund large-scale sequencing. They're often targeted to specific disease areas and not really a health system-wide funding. So that was never an option. And insurance companies, sure, they will pay increasingly genetic testing, but the way the reimbursement works and what gets reimbursed is often not the comprehensive genomic test. It's often not the whole genome sequencing test. It's different panels, and we know that then that'll lead into delay of diagnosis when you do these multiple layers molecular tests that are reimbursable but are not the conclusive test. So we chose also to raise awareness within our community and find we were lucky to find a large number of very generous donors that want to support the advancement of genomic medicine. But the overall goal within the next few years is that we do exactly what was discussed, show to the payers that this is the way forward. We should do the comprehensive test as early as possible to get the diagnosis and save money and save the agony of the families. And that's a good investment from the insurance company, and they should pay for going forward.
No, thank you for that. You know, the next session after this one is on how health equity can be improved, you know, in the era of precision medicine. I think Tomi, you and I think as well as Peter and Josh brought up the fact that access to genomic technologies, comprehensive sequencing and beyond is not universal. And that there remain, as we know, not just for this, but for many things within our healthcare system, large populations that are underserved or underrepresented, including people at or below the poverty level and underrepresented minorities within our population. I suspect that it's the sort of the barriers that we're familiar with for access to care in general. But two-part question, and I'll start with you, Tomi. Are there barriers beyond the standard sort of access to care issues related to insurance, transportation, and the like for underserved minorities or underrepresented populations? That's one. And then the second is, how are you at Kansas City, handling this? And love to extend that question to Peter and Josh as well.
So I wish I would have a perfect solution for this problem, but it is a problem that we identified in the first 2 years of recruitment. We started to compare our patient population that entered the sequencing program, which pretty much represents the patient population that enters our hospital, in at least in the specialist clinics. And we saw that we had gaps in the inner city minorities as well as then in rural populations. So those are the two challenges that we're trying to tackle now. And our initial approach is to embed patient recruitment within the clinics that see the underserved populations and minorities, and address some of the issues that are kind of not that common for us, to not being able to reach a family once they've visited the clinic, so that we do an intervention early on when the physician first suspects that there's something wrong, so that the opportunity is not missed because the connection with the family is missed or something. So kind of new approaches into having folks in the front lines and engaging the families is a key thing. But I think there's a lot of issues related to genomic medicine in general, and potentially lay populations suspicions on what genomic medicine is. And I think that's an underexplored area. And I think what we need to do beyond going to these primary care clinics is to participate in community, in faith-based communities, in different areas of the community, and talk about genomic medicine and what it means, and mitigate the fears that may be unfounded in population.
It's very helpful. Peter, Josh, any thoughts?
Yeah, I think those points are great from Tomi. I think we've struggled with how to overcome both access as well as trust with the medical establishment in various groups that have historically been kind of mistreated. I think as kind of a segue, to think about how can we solve this problem, that in the next 5 or 10 years, I don't think we're there today, but if you look into the future, what's a way that we've helped diagnose treatable disorders, across a large population? And I think the example of that is newborn screening, which has been hugely successful historically. If you think about the past 50 years, newborn screening's at this crux right now where they've developed a biochemical test for newborns that's treated the diseases that we knew how to treat 20 years ago. Now we're at this point where we have all these therapies coming down a pipeline. If we don't diagnose people in time, we're not going to get them the therapy, but then we can't keep adding biochemical tests to newborn screening. So I think we're going to see this kind of congruence where maybe we need to think about how do we use newborn screening, which is something we can do at a population level, overcome some of these disparity problems with diagnosis. And then hopefully that's also a way to kind of tie in patients and families directly into the medical system if we can make a diagnosis in a timely, quick fashion at the newborn screening step.
That's perfect. Thank you. I was thinking about newborn screening, earlier as the discussion was going on, and I didn't necessarily want to go there, but it seems seems like a logical extension to me, and I would hope that in 20 years from now that heel stick yields enough mononuclear cells to allow for sequencing. We can all dream. I do want to, we have only a couple of minutes. I do want to sort of end on a note that I think is forward-looking. Myself working in industry for the past 13 years, I always try to think about industry as a group of professionals who are looking to help solve critical problems by applying capital and scale. I'm curious to hear from each of you briefly in the last couple of minutes, and maybe Peter, starting with you. What is the role for the diagnostics industry, for the therapeutics industry, for our colleagues in the IT industry, and I think maybe as an overarching group, the investor community who ultimately provides initial capital for new projects here. How can industry help in this, to advance the work that you're doing and make sure that no child is left unsequenced when they need it?
No, and I think like some of the partnerships that we've worked with, like working with industry groups that really have the expertise in like patient phenotyping and working with the electronic medical record. I think that's an example for us that's been like, hey, these companies have developed some very sophisticated products around being able to take that information, and like we have the expertise in the genomics, and sort of working together to tie that tie that phenotypic information back to the genomic data. That was the first thing that came to my mind, but I'll let my colleagues answer that.
Josh, you want to go next?
Sure. I think I guess there's 2 points I'd make. One is that I come from a very academic background, but when I think about some of the opportunities here, we're really at this threshold of totally transforming the way medicine is delivered in pediatrics. We're not really we haven't been in the dark ages, but we are just at this point where there's a huge opportunity. So I really encourage everybody on this call to jump in, because it's such a huge opportunity. The second point, and the reason I brought up my kind of academic background, is that historically I've always been had a more nervous thought process around industry. But if you think about the genome sequencing project, the first human genome back in the early 2000s, where NIH had kind of this very nice concrete approach to how they were going to do it. But then with the entry of Craig Ventner into the field as a private with an industry approach, I think that galvanized the NIH, as well as Craig Ventner's work, and I think having kind of almost like having 2 Manhattan projects going at once, made the Human Genome Project happen in a faster fashion, and then has brought us to the point where we're having this discussion today, where like instead of costing $2 billion to do a human genome, we can do a human genome in a month, or a week, really, at less than a couple of $1,000s. So I think industry plays a key part, and I think there's opportunity for all of us to work together on this.
Tomi, I'm going to give you the last answer to the last question before I wrap up.
Yeah, so I agree with both Josh and Peter on this. Industry has a huge role to play, especially with the cost currently. Even though genome sequencing cost is going down, it's still costly, and the data is extremely costly. So I think what we need to bring onto the table is some of less common players in field of precision medicine, which are becoming more common, like Google, AWS, Microsoft. They are really the key people and technologies that will enable scaling the genomic medicine to every child, and we need their help beyond the electronic medical record providers that also should take genomic medicine more seriously and have methods to integrate that into EMRs. But I think we need the big technology companies to understand that the future will be the newborn screening by whole genome sequencing.
I couldn't agree with you more. It's almost as if you can see my notes here, because I've written down, Microsoft, AWS, and Google, amongst others, as potential engines of innovation here. I think maybe just in summary, and really to close, what for me has been a fantastic discussion, and for the broader audience. Tomi, Peter, and Josh and I had a pre-call a few weeks ago lasting an hour, which was, by the way, completely different set of topics here, all of which equally detailed. So, for me, 2 hours of this is not even close to scratching the surface of, how rich this area is, the idea of extending precision medicine throughout pediatrics, helping to provide diagnoses, to uncover new areas of biology and new biological insights. Hard for me to believe that that the good here is limited to just ascertaining a diagnosis for families and patients in need, and resolving the unknown. There are new insights into human biology to be gleaned here from germline disorders, many of which manifest during childhood, the potential for therapeutic development, and really the potential to affect cost savings throughout the healthcare system for children, who sometimes can be trapped for years, in search of a diagnosis, or sometimes going down the wrong road. So there's a lot of good to be done here, a lot of benefit to be had and created for patients and families, a lot of value to be created for those who want to work in this field. And I could not have enjoyed this discussion more. It's been very thought-provoking for me personally, hopefully for all the people in the audience. And I really want to extend my deepest thanks to Tomi, Peter, and Josh, not just for being participants in this discussion, but really for being at the cutting edge of something that is fundamentally important to patients that we all care about, and hopefully we all includes everybody listening today that is children with unmet medical needs. And so with that, we are at the stopping mark, and maybe I will turn it back to Stephanie. And thank you again for, and to the MedCity News staff for inviting all of us to participate today.
Well, thank you, Sam. And thank you to all the panelists for a very compelling conversation. I mean, you only had an hour, and yet it was such a beautiful balance between talking about these cutting edge technologies, and yet the sort of the personal journey that patients and their families are making through what can be a very kind of frightening, frustrating period of time.